Hello, I'm Jim Cornell, and this is Base to Base, a weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 25, going out on September the 5th. That is, in case you weren't aware, International Day of Charity, which is a great idea. It's Samosa Day, the day Voyager 1 went into space, and it's the birthday of Tolstoy, Jesse James, and Freddie Mercury. And there is a Freddie Mercury connection to this week's interview. Intrigued? Well, hopefully you are, and hopefully the conversation will rock you, although not literally. It was my birthday last week, and for the gift for the man who has nothing, my family got together, and without saying who it came from, someone gave me COVID. A very generous gift. My voice hasn't quite recovered yet. When I was on the radio all those years ago, when you had to wind them up, I always got calls saying how good my voice sounded when I had a cold. Not sure if that's a good or a bad thing. It's been even busier than usual on the news front as the summer hiatus seems to have ended, so I still have a few articles left over for early next week. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us or in sponsoring a podcast episode. Last week our conversations were a very topical one on the effect of cuts to funding in the US with Jean Pécu, GenoFab founder and professor at Colorado State University, and that one went down well. A few comments on that one. And we also chatted about the fight against Parkinson's disease with Antti Fuolanto, CEO of Herantis Pharma. This week it's an in-depth conversation on molecular glue degradation with Monte Rosa Therapeutics Chief Data and Information Officer John Castle. Just the one this week because it is a bit longer than usual. First, let's take a quick look at the news you may have missed over the past week at deeptechdigest.com. A molecular stopwatch measures the tails of cell RNA. The FDA approved an early Alzheimer's disease treatment, and IQVIA and Flagship Pioneering are going to collaborate on developing breakthrough life sciences companies. No mention about which company is providing the sandwiches, though. Speaking of collaborations, there's one looking to advance multi-omics innovation in South Korea. Hongene is going to supply oligonucleotide technology under a new licensing deal, and menin may be a new target for bladder cancer. Rayner Biosciences has unveiled a generative AI platform for mRNA therapeutics. D3 Bio has received a KRAS G12C mutated cancers boost, and Cage Bio is collaborating on treatments for cutaneous graft versus host disease. Intron Bio has filed some gene editing patents. Personalis revealed new data from its lung cancer trial, and you can read all of these and plenty more at deeptechdigest.com. So let's move on to this week's conversation, and it's a discussion on molecular glue degradation with Monte Rosa Therapeutics Chief Data and Information Officer John Castle. We cover the use of machine learning and AI on undruggables and the company's recent cover story in Science. And, as I alluded to at the beginning, Queen. It would have been Freddie Mercury's birthday and Queen come up in the conversation. Really? Is it synchronicity? Well, no, that was by the police. It was more like a kind of magic. And I'll stop there and we'll get on with the science. Hi John, thanks so much for being with us on the podcast, and to get this underway, maybe you could give us a bit of background on the company.
It's great to be here. Thank you so much for inviting me today. I'm going to start, I think, a good place, maybe, my background that can give you, I think, a good setup and the foundation for what we've been doing at the company. Growing up, I loved math, I loved physics, trained as a physicist, and insofar as, first degree in physics, and then working in geophysics, actually seismology, believe it or not, doing a postdoc at MIT in Boston. today it would be called a data scientist. I was pulling together all sorts of information, working with other professors, different colleagues. My specific focus was looking at the structure of the inner earth, the core mantle boundary. But at the time, I was really getting excited for, when I learned about what was going on in biotechnology, and this was around just before 2000. There was a small company out in Seattle started. It was called Rosetta Informatics. A guy named Stephen Friend had started this. And it ended up being the first company that was really acquiring large data sets, transcriptomics, other omics. And Stephen had the foresight to hire, there was about a dozen physicists that he hired because the molecular biologists were incredible, but they, unless you could analyze the data in Excel, they couldn't make heads or tails of it. We were really, I think, the first biology quants, if you were. They were coming in and being able to take large data sets and doing something useful with it. I had a great run there. 2009, want to apply these tools, these omics tools, to the immune system. Found someone in Mainz, Germany, Ugur Sahin, who was just about to start a new company, and I joined up with him, and we founded biotech back in 2009. Had a great run. A lot of the first vaccines, the neoantigen vaccines, using the RNA platform. Got me very interested in the immune system, TCRs, vaccines, what you could do with bispecifics. A few years later, Marcus Varmuth approached me and said, "Hey, John, have you ever thought about what goes into the proteasome? How you could use small molecules to modulate the protein degradation?" Honestly, I knew very little about small molecule chemistry. I knew very little about structural biology, and it's been just amazing. When it started, I think you got to take three different concepts and integrate them. So the first one, there's a ton of targets that are well validated but never could be targeted. These are undruggables. I sometimes call them the always wanted, never could targets. They've got really great validation from human genetics, from pathway studies. People have started to approach them with different editing techniques. CRISPR being one, editing DNA. RNAi, editing the transcriptome. Now, great technologies, but man, if you look at CRISPR, it's really difficult. Some of the therapies that have been developed and approved are millions of dollars for each patient, so very expensive, laborious for the patient. Now, that's number one. Number two, I'll just give you a hint where we're going, that there's a system in every cell in our bodies that's called the ubiquitin proteasome system. And what this is, it's a natural trash collection or recycling mechanism where proteins called E3 ligases, one of them being cereblon, actually tags proteins, sort of like a kick me sign that it puts on the back of them called ubiquitin. And with this marker, they're tagged to go into the proteasome, and the proteasome is, if you will, the chipper or the garbage disposal. It cuts up a protein with a tag into small pieces. Now, part three, there was this drug approved in 2006 called lenalidomide. Incredibly successful financially, was a blockbuster because it was helping so many multiple myeloma patients. It was really game-changing for multiple myeloma. But in 2006, and remember this is before iPhones, before all the technology we have today, no one knew the mechanism of action, the so-called MOA. It just worked. And so there was no way to know how to leverage this mechanism because we didn't even know what it was. Now, fast forward to 2010, 11, 12, and I think you can guess where we're going. It was understood that actually lenalidomide binds to one of these tagging proteins, cereblon, and changes or morphs its surface so it tags a transcription factor called Icaros, or IKZF1, that's essential for multiple myeloma cells. And what it does is it takes it out, it edits, it degrades the protein, and so we've got now proteome degradation, and the multiple myeloma cells die because they need this protein, this transcription factor. So it really puts those three pieces together. Can you edit the proteome with a small molecule, and using an endogenous system and proof of concept with this drug called lenalidomide? And that was the premise of Monte Rosa. Can you use this understanding to develop new drugs?
Moving on then, I wonder if you could give us a bit of a definition of molecular glue degradation.
Yeah, no, I'd love to. So what really the concept is, can you induce a protein-protein interaction between a tagging protein such as cereblon, and there's dozens of these in each cell, and modify its recognition of what's called substrates, or the proteins that it's binding to, so that you've redirected it to tag disease-critical, disease-relevant proteins and edit them out, degrade them from the cell. And so what you've done is used a small molecule to induce the PPI, the protein-protein interaction, and removed or degraded the protein.
And so what's the role of cereblon?
Yeah, so cereblon is really, it's part of a complex. There's many different proteins. There's actually an E1, there's an E2, and there's an E3. And this E3 ligase is the tagging protein. And so it has a molecular recognition. It has a surface that then binds to a target protein. Sometimes I think of it like jigsaw puzzle pieces. Obviously, it's not just one-dimensional or two-dimensional, but in this case, with proteins, it's six-dimensional. And so what you're doing is trying to make sure that you've modified with the small molecule the surface in just the right way that it's warped to recognize this disease-critical protein. I mean, this is not easy. Let me just be clear. It's really difficult. You've got all sorts of challenges that goes along with this. You can think about all the history of small molecules. And when we started Monte Rosa, many of the investors said, lenalidomide's there, this proof of concept drug, but glues, they can't be selective. They can only be found serendipitously. And the target space is really small. And so we had to contend with that as we started the company, is because that's what it was at that point 6 years ago. And so what we did was we took some time, we put together an amazing team, amazing resources, or I should say capabilities, and a platform that's just driving us forward. We put together a team, a lot of chemists, cutting edge chemists, a lot of profiling technologies, high throughput screening. We've got so much proteomics. We've really invested deeply because we're editing the proteome. Structural biologists, X-ray crystallography, cryo-EM, and at the same time, a lot of algorithms also. So we put this whole team together to solve these problems and be able to make these drugs going forward.
And so what's the connection between that and treating disease?
Oh, so this is also where I think a lot of the AI comes in as well. There's multiple parts where we're using AI. One is to really understand what are those critical proteins that haven't been drugged to date. They're undruggable, but are essential for diseases. And you can think if you could take them out of the cell, are you alleviating disease? And we'll get into later some of the targets that we're going after specifically and how they're helping patients. But that's one aspect, is using a lot of information, everything from LLMs to genetic-wide association studies, UK Biobank as one example, to identify proteins where if you remove them, you benefit the patient. If I may, I'll go on a little bit about how we're using AI throughout the whole platform, because the next question is, which of these proteins that you'd like to target have a surface that's amenable to binding to this E3 ligase called cereblon that we've been talking about. Again, I mentioned PPIs. And so you have to be able to form an interface that's attractive. And that's really something that we've had a breakthrough there, because I think a lot of the field is still very focused on protein structure and figuring out how a protein folds up into a ball. And that's important. But the next step is to figure out what's the profile on the outside and what's the surface, because that's what's mediating the interaction between two proteins. And you can have many different structures that have the same profile. And so it's that protein that profile or that surface that's mediating it. Then we're using, along with our data, our big data, we're using a lot of AI to identify what is the small molecule that's going to induce that warping in just the right shape. Warping of the protein is going to induce the right action and cause that stickiness, that glueness together. Then finally, once we have the right small molecule that's selectively degrading the target protein, we're using a lot of algorithms to figure out what are the right patients that are going to be addressed. Is this all lupus patients, or is it a specific subset of lupus patients? Is this all ovarian cancer patients or a specific subset of ovarian cancer patients as well.
I guess, though, when you are into the details of degrading a protein and stopping something, there's not necessarily a guarantee that that is going to be something that helps. I mean, it could be that it either reroutes itself or it does something else, or there are side effects, or I guess that's not the end of the story, though, is it?
No, you're absolutely right. I mean, the advantages of glues are incredible. I mean, we've got oral molecules. They're very selective. Once they get into the cell, they act catalytically. We're not just inhibiting a protein, but we're actually inducing its removal from the system. One small molecule can induce the degradation or removal of many, many copies of the target protein. Now, to your point, once that's gone, is that going to be benefiting the patient? And certainly we're doing a lot of work to identify the targets that we believe going in are going to be beneficial. Many of them, and I think one of the huge attractiveness of this modality, of this platform, is that we're going after new targets, novel targets. But that certainly has an element of risk because they're new targets. And what is the downstream biology of that? So we have an absolutely fantastic biology team that then rapidly tests those in different systems to see, is it doing what we expect? Starting off at a cellular level, then moving to animal models, and then certainly as we move forward into more standard tests to make sure that you don't have any metabolites, for example, from your small molecule or anything else that may cause a problem. And then very excitingly going into clinical trials in humans as well.
Next, then, could you tell me a little bit about the Queen?
And,
But not the band.
We have a lot of platforms. We have our Bohemian Rhapsody. We have, a lot of, we have Glue Aid. We have lots of different platforms that are named after bands, particularly around Queen. Our CEO loves the band. We give away T-shirts all the time. It stands for Quantitative Engineering and Elimination of Neosubstrates. Just by accident, of course. No, so the Queen platform is really, it's our discovery engine. And we've brought together all the technologies, the people, the experience to come in with, okay, let's say we want to go after a target. How are we going to approach it? And there's not a one size fits all. We have a ton of proteomics data in what's called our data moat, where we're already testing molecules in different cell lines and seeing how they behave. We can then start a virtual screen campaign against that target as well. We have the capability to do high throughput screening, both biochemically and cellular assays that we've put in place. And then once we start that, then there's a certain cadence we have for then modifying the molecules, seeing how they perform, and then running that again and again and continuing to improve those. And this all happens within Queen. Certainly a lot of algorithms, computational chemistry, screening, proteomics, all contained there. The group really functions. It's a high-performing machine and delivers out leads at that point that then go into fairly straightforward lead optimization campaign, where you're then already starting to think about everything from CMC to DMPK, ADME properties, and starting to look forward into what is your patient population going to be. But Queen really is our name for the sort of the umbrella concept of the discovery assays that are showing us how to get those first hits and leads.
So you're not using mercury in Queen or anything.
all of our meeting rooms are named after Wembley, are named after different concert sites. So it runs deep.
You mentioned AI earlier. How has that assisted in the process, and how does it continue to be employed in the company?
It's just fabulous right now. I mean, we're using it throughout, and if you will, some of the more mundane ways, but I'm really encouraging the rank and file. How do we leverage these tools for writing documents? And so this is something where how can we just get faster at what we're already doing? That's well underway. It's very useful. At the same time, I think some of our more, if you will, cutting edge or usages that are very specific, we have what we call target aid, where we're really incorporating all sorts of information into LLMs, whether it's from PubMed, whether it's from abstracts, whether it's from SEC filings, whether it's from clinicaltrials.gov. All that information is parsed and then at our fingertips to know what are the hot targets, the hot opportunities we should be looking at. This concept of, and I'm going to mention it again, but the protein surface and the neosurfaces is so powerful because this gives us, I think, a key shift in the way that drug discovery is going is in inducing PPIs, these protein-protein interactions. And it's no longer having a small molecule that just sticks to a kinase and then inhibits its activity, but it's really a small molecule that's, if you will, mucking with a system that's already there and modulating how different proteins interact. Without the algorithms, we'd really be guessing, but with the algorithms, we've got a good starting point as to how two proteins can come together and form a complex induced by the small molecule. Conceptually, it's this idea of surfaces and how do you leverage that. I hired a guy, Pablo Gainza, who really rewrote the book on how you look at protein surfaces. He used geometric deep learning, some ideas that came out of there, going beyond just GNNs, and able to characterize protein surfaces with 6 different proteins, certainly the XYZ coordinates, the curvature, but polarity, hydrophobicity, and so that you have to make complementary surfaces, or in some cases, surfaces that mimic each other. And that's all done through algorithms. Then once you have a potential PPI between two proteins, how do you fill that space, if you will, between them? What's your small molecule going to look like? There we've had an incredible success with virtual screening. We've been able then also, once we can predict how does a small molecule, what's its shape? Something people talk about the SMILE structure, and this is all 2D, but obviously a small molecule has a 3-dimensional shape, and how does that fit into the protein? And we've been able to successfully predict the shape and how it's going to warp the surface, and so that's further helping us both with virtual screening, but also hit expansion. We worry a lot about ADME properties, this whole multiple parameter optimization, MPO, and how do you keep your small molecules that it's going to be oral, it's going to be absorbed in the body, it's going to have the right PK, it's going to be in the body the right amount of time. And there, within our chemistry space, we have amazing models that are very predictive for the ADME properties. And so as we develop the molecules, we're able to keep them very drug-like and not get off into, if you will, never land of drug space where, if you're not careful, you can go off into a wacky drug space it's not drug-like, because you have to keep something not only active, but also very drug-like. And we've been able to do that very successfully. So it's really putting these pieces together. I think of them as individual modules that we can reuse in many ways, the same sort of modules that we use for lead optimization and keeping these ADME properties very drug-like. We also use them when we expand the library.
We always hear about successes and how things are working, but what have been some of the challenges in this space?
It is challenging. And first of all, obviously, you need a great team because, as I point out, when we started the company, the people said, hey, it has to be serendipity. There's no rational design. They can't be selective. The target space is small. I'd say one of the challenges then is how do you get outside of that? I can say we started off with some projects that were sort of following the beaten path, spent some good time, learned a few things. I think we were fairly smart that we parked some of these programs early on just because we realized the biology didn't support some of these targets. And so that's one point. Back to some of your earlier questions, when you go into the undruggable space, you really need to be willing to cut your losses at some point if the target doesn't pan out, or if you start to see some issues with degrading the target. Another big challenge is just that what's called the SAR, or structural activity relationship, is very sharp. I mean, there's cliffs everywhere. And so if you just, in some cases, change one atom here or there, you can completely get rid of your activity. And so understanding that and being able to accommodate that and be able to react, because we have gotten into places where there's been dead ends in terms of some of the development projects. So that's been another challenge. Yeah, I think those are some of the first challenges that we've met. I think we've been able to overcome them as well, but certainly it makes the bar high for moving into the space and working in the space.
How broad is that target space, and how do you prioritize targets?
This really relates to the science paper as well, is that the dogma in the field was that you had to have a certain protein structure on the outside, exposed on the outside of your protein in order to interact with cereblon, the E3 ligase. This was called a G-loop degron, G being glycine, and glycine had to be at position 5 in this loop, a little bit like a piece of Velcro, if you will, or a latch on a jacket. And if you didn't have that structure, and so the thought was, okay, you have maybe a few kinases, you have a lot of zinc finger transcription factors, and that was the target space. What we did in the science paper, and this was a great snapshot of our platform of Queen about 3 years ago, was to show that if you have the right chemistry and you've diversified the chemistry, you can actually blow that paradigm away entirely. You can warp the surface of cereblon in different ways, in diverse ways, and engage very different target spaces. We started off by showing that we could predict which proteins have these G-loop degrons, these canonical degrons. We then showed that if you modulate and diversify the chemical space, you can engage proteins that have different structures, that it's not just a canonical G-loop, but there might be a helix instead of a beta turn with the G-loop. And so that further expand the target space. Next, back to the surface concept, was that you actually don't need to have a G-loop degron at all. You just need to have a surface profile that looks like or mimics a G-loop degron and can be a very different structure. That was the VAV1 target. There's no G-loop structure, but it mimics the surface very nicely. Now, one of the things we haven't published is that with the right chemistry, you don't even need to have something that looks like a G-loop degron. So you can go very much beyond that. And with different chemical classes, you can engage very different proteins with different surface characteristics on the outside as well. We're continuing to expand the target space. And so it's not just constrained to a few zinc finger transcription factors, but it's much, much larger than that.
What's the reaction been like to that publication that you had in Science?
It was fantastic. And we thought about it for a while, do we want to publish this? And we thought, okay, yeah, we do actually. We want to put the stake in the ground to show, what we've been able to succeed, where we're at as a company, and where the science has taken us as well. The point of the paper was that we can break this paradigm with the right chemistry, and we understand how it works. We have algorithms, we have the platform, we have the data to understand, to propagate this knowledge, and with the algorithms take the understanding and run it across the whole proteome. The reaction's been fantastic. You don't have a paper on the cover of Science every week, and so it was just very fulfilling. But the feedback, breaking previous paradigms, a tour de force that would have been impossible earlier, fabulous manuscript. My experience as a reader was full of great excitement. I mean, this is just what we heard back from different people. The feedback also from colleagues from within biotech, within pharma, actually investors also, really understanding what we do and where we're taking the platform. What was nice also, as I mentioned in the beginning, the questions about serendipity versus rational design, about selectivity and about the target space. And this paper put those concepts in a coffin, deep six the question, do you have to use serendipity? No. We showed you could do this rationally. Could you be selective? You can look at some of what's called the volcano plots, where we've profiled the molecules in unbiased proteomics. One protein is being degraded, and only one protein. And finally, to your earlier point about the target space, we showed how it really, this blows it open and is much larger. So it was really great and to get that feedback and to really put the stake in the ground.
Could you tell me a bit about the phase 1 study and the timeline for that?
As a scientist, I love to talk about the science, but at the end of the day, really the point is to make medicine. And that's where we're at right now. 3 clinical trials running as we speak. The most recent one is MRT 8102. In our society today, there's a lot of talk about chronic inflammation, and it's a problem. It's scary. I mean, how it's affecting us every day. One of the things, as one example, is cholesterol. I didn't even know until recently that cholesterol forms crystals within our bodies, and these crystals can be needle-like. This is part of atherosclerosis and other diseases as well. They can, when they get into cells, often monocytes or macrophages, then they can blow holes in the lysosome, unfortunately, releases caspases, and you get this whole series of events leading to something called inflammasome being activated. In particular, the NLRP3 inflammasome is activated. Downstream of that, it induces or activates the secretion of IL-1 beta, also IL-18, both in this case pathogenic. IL-1 beta leads to IL-6, leads to other states as well. This can be useful if you have acute inflammation because of some trigger, but in this case with cholesterol or other crystals can basically lead to chronic inflammation. Now that NLRP3 inflammasomes really received a lot of interest. Many different companies are going after it, trying to figure out the best way to modulate that. Part of that complex, the NLRP3 inflammasome, is a multi-protein complex. One of the proteins that's essential for that is NEX7. What 8102 does is to very selectively degrade NEX7, leading to a lack of function of the NLRP3 inflammasome. And in preclinical models, we see a very nice decrease in IL-1 beta, IL-6, and other cytokines, also of IL-18 as well. The trial started in July. It's a healthy volunteer trial, so a SAD-MAD trial. There's also a part 3 in which we'll be enrolling patients with high CRP levels. We're guiding to a readout in the first half of next year. Really exciting. We think this is going to be a great modality, oral small molecule that's going to benefit these patients.
And as if that wasn't enough, what else do you have in your pipeline?
Yeah, staying with immunology, we're right now planning phase 2 trials of a drug called MRT 6160. If 8102 and NEX7 is inflammation, 6160 is autoimmunity. It degrades a protein called VAV1. VAV1 is another one of these always wanted, never could targets that's downstream of TCR and BCR signaling, so in T cells and B cells. It's essential for Th17. It's also what we see when we in preclinical models is a decrease of cytokines such as CD69 and really a dampening of this autoimmune relevant activity. Great preclinical studies. Novartis actually came in and we've partnered with Novartis on this. We are working with Novartis really effectively as we plan the phase 2 studies there. We also in the immunology and inflammation space have many additional unclosed targets that we're working on as well in the Queen platform. Switching to oncology, we have an ongoing study with 2359, MRT 2359, where we've shown and published activity in prostate cancer. That's continuing. In the cell cycle, we have degraders for cyclin E and CDK2. Those programs are moving along as well. And then other targets coming behind that. So really a full portfolio and pipeline.
Right. You mentioned the partnership with Novartis. How important are partnerships to you?
They've been fantastic. I mean, our first one was with Roche. I work in our Basel office, and coming into Basel, you've got the Rhine, you've got Roche on one end, and Novartis on the other. Our collaboration with Roche has just been fantastic. That is focused on oncology and neuro. Obviously, Roche is a world leader in both of those areas. Working with them has really helped us turbocharge some of our work, and those are continuing. The deal with Novartis was specific for MRT 6160. What we see there is that they have amazing resources and capabilities in running later stage trials, phase 2, and going toward commercialization. They can do things, multiple trials in parallel. And so just super excited as we think about the phase 2 trials, of 6160, how they can do that in parallel and move things forward toward marketization and commercialization so much faster than we as a small company could. So it's been very instrumental and fantastic.
Are there a lot of companies working on molecular glue degrading?
There are. It's a big space. Certainly Roche and Novartis. Also, BMS has been working in the field. Several small companies, some companies working on PROTACs, which are a related molecule, a little bit larger, different target space with those. They're certainly having some successes as well, which is great to see. We see a few companies coming into the molecular glue space, several academic groups, and there's other modalities in terms of trying to not only induce degradation, but modulate PPIs either with different types of antibodies on the surface or different small molecules that might be, for example, inducing lysosomal degradation rather than proteasomal degradation. What I think we're seeing right now is the rebirth of small molecules. Small molecules, maybe it was your father's Oldsmobile, so to speak, and wasn't quite as exciting as some of the large molecule programs. Now I think we're really changing the narrative, and we're seeing with small molecules, you can do things that people 5-10 years ago never thought possible. And the advantage is then, once you've put in the work to develop these molecules, it's an oral molecule that you take. You can take it at home. It's very convenient. If I think back to our company, I mentioned, can we do this rationally? Yes, it's not serendipity. Can we make selective molecules? We understand the system, so the answer is yes. And then the target space, we've gone beyond these G-loop degrons with the chemistry. One of the things I'm so excited about is that while cereblon, we talked about cereblon is our workhorse ligase, we've already started applying our knowledge, our platform, our processes to other E3 ligases and further expanding the target space as well. And so the opportunities that we're seeing are just enormous. We're certainly, I think, in a leadership position right now, and it's exciting to see how the field will continue to develop both at Monte Rosa and at other companies in the area, both in terms of pharma and biotech. So long answer to your question, but a lot of activity in the space. And I think it's really, really warranted given the potential of this modality.
As I mentioned earlier, just the one interview today because it was a bit longer than usual, but certainly well worthwhile. And some amazing work clearly coming out of Monte Rosa. And hopefully now you all know about molecular glue degradation if you didn't before. And now you also need to name all of your meeting rooms and equipment after your favourite band. If I was in that position, I would have a very hard time choosing. The Beatles is an obvious choice, but I think I'd probably go a bit more obscure than that. Let me know if you have some similar quirks at your company. I'd love to hear about it. So that's it for another podcast. A wet weekend here again, and it doesn't really matter anyway because I'm still recovering from COVID. But it's also the air show, so there will be lots of planes in the distance. I'm not really that bothered about it, but I would enjoy seeing the Lancaster Second World War plane flying over. I have seen them plenty of times in the past, but it's still a nice sight. The Lancaster does have a nice, unique sound, although it's really got nothing on the Vulcan, if you've ever heard one of those scream over your head. Not sure how we got sidetracked there, but it's pretty easy for me to do. If you have questions or comments, suggestions for guests, or want to sponsor one or more podcast, please do get in touch, and you can do so at jim@deeptechdigest.com. I think we're already booking interviews into October, so that's a good thing. So until next week, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next week for another Base to Base Biotech podcast. Now, where did I leave that Queen's Greatest Hits CD? The Base to Base Biotech podcast is an Air Coastal Media Limited production.